So welcome back to our fourth webinar of Immunoassay Applications Week. We've had live workshops every day going over best practice for different immunoassays. It's been a really popular week so far, really engaging conversation and lots of great questions from the audience. So hopefully we can continue that today. My name is Dr. Poppy Nathan, and I am a scientific marketing specialist here at Proteintech, and I'll be chairing this workshop. And I'm really excited to have Rahul Panchal with us today, who is a product manager here at Proteintech. So as I mentioned, we'll have a Q&A session at the end. So any questions that you do have, please just pop them in the chat or in the Q&A box, and we'll get onto those. But first, just a quick introduction to Proteintech, in case you are unfamiliar with who we are. So Proteintech is a leading life science reagent manufacturer. We were founded in 2001, and at that time, we were a primary supplier of antibodies. So we now have antibodies against over 14,000 human proteins, which covers about 2/3 of the total human proteome. We also offer immunoassay kits, including ELISA kits, IHC kits, cell separation kits, and antibody labeling kits. But since then, we've expanded. Our portfolio now includes more product lines. So we have our humankind branded range of recombinant proteins. So all of our cytokines and growth factors are expressed in HEC293 cells. So you can be confident that they'll have the native folding and post-translational modifications and glycosylation that you would expect to see in human proteins, which ultimately increases their bioactivity compared to proteins raised in systems such as E. coli. Importantly, we can manufacture all of these products at a GMP grade for use in cell and gene line therapies. So we also have our Chromatech branded nanobody-based reagents, and nanobodies are about 10 times smaller than traditional antibodies, and so they can bind to their targets with really high affinity, which makes them ideal for IP, super resolution microscopy, and protein purification. And I'm sure Rahul will go into some of these products in a little bit more detail for you today. And finally, we have our genomics line, which is a subsidiary of Proteintech. And here we manufacture antibodies conjugated to oligonucleotides, which allows you to perform protein quantification during your RNA sequencing. So all of our award-winning products are self-manufactured. We actually have the largest portfolio of antibodies for the human proteome, and we have a rapidly growing recombinant portfolio. For convenience and for testing, we offer most of our antibodies in a 20 microliter trial size. We also really understand the importance of high-quality product validation here at Proteintech. So whenever we're testing new products, we validate with relevant sample types, appropriate experimental controls, and we try and use knockout and knockdown models wherever possible. And all of this validation data is completely open access to our customers. You can find it on our website. And we are a company of scientists, so we have a very in-depth knowledge of immunoassay applications, and we can offer outstanding technical support for all of our customers. We have 7 inventory sites, and we have manufacturing and research sites now on 3 continents, so we can offer next-day delivery to many countries across the globe. We define our success as a company through our customers' scientific success, specifically through helping you publish impactful research faster. We have over 350,000 citations now globally, which makes us in the top 4 cited antibody companies. But I also just want to highlight that as a company, we strive to make the searchability of our products as user-friendly and informative as possible. So we've actually launched some new validation initiatives to help our customers select the right products. So we have experimentally mapped our recombinant antibodies, and we're sharing that information through our unique 3D epitope viewer. So this is a tool that allows you to interact with epitope structures and explore antibody binding sites, so you know exactly where your antibody binds. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen selection function. So this is a toggle that allows you to filter specifically for antibodies generated from your region of interest. And additionally, you no longer have to filter through all of the variations of an antibody from the search results. You can view all formats, including conjugations, buffer formulations, on just one page, which ultimately is here to help streamline the process for antibody selection for you. And very lastly then, I'd like to highlight that we have built off of the information from our epitope mapping in our database, and we've recently launched an AI-powered search tool on our website called Able AI. So Able allows you to directly chat and ask for product recommendations, help with experimental design, and access relevant validation and citation data. For example, you could ask Able to recommend you specific antibodies validated in this specific sample type, or you could ask for specific experimental protocol advice. For example, can you suggest optimal fixation and polymerization products? And Able will be able to find you the best product for your experimental needs so that you don't have to spend countless hours searching for the right antibody. So Able is a first of its kind tool designed by our scientists in-house with customers in mind. And it's really made so that you can focus on your discoveries and not spend hours on reagent selection. As I say, we're very proud to be the first in the industry to launch anything like this. So do check it out if you've got some time. You can access it either through the QR code on the screen there, or you can find it directly on our website homepage. And then with that, I just want to say a massive thank you to Rahul today for coming on to talk to us everything IP. So Rahul is a product manager here at Proteintech, where he works to grow product portfolios through strategic product positioning and sharing through targeted marketing campaigns. He's also responsible for creating new content to help present the usage of these products against various cancer research areas. Rahul has a master's degree in biomedical sciences from the University of Connecticut, and he has over 8 years of lab experience at the bench, working for numerous biotech companies in research and development. So he really is a bit of a master at all things immunoprecipitation, and will be able to answer all of your specific questions today. So with that, I just want to say huge thanks again to Rahul, and I will stop sharing my screen and let you take over.
Okay, can we all see the screen here?
We can see your screen. It's not in presenter mode if you'd like to.
Okay.
If you can.
How about now?
It still just looks like a normal PowerPoint presentation as opposed to the presenter mode.
Well, it's open on a slideshow on my computer, so it's strange. Okay, 1 second. All right. Does this look better?
Yes, perfect. There you go.
Yes.
Thank you for
Okay,
that.
great. All right. Well, thank you, Poppy, for that introduction. So yes, as she mentioned, we are here today to talk all things immunoprecipitation. Let me just turn on my pointer. All right. So, for this talk, we will look at a couple different things, okay? We're going to talk more about what exactly immunoprecipitation is, what are the steps involved in the process, and how you can optimize all those steps to get the best IP. We will look at criteria you should consider when selecting an antibody, as well as a matrix for IP, when perhaps you could use a nanobody instead of an antibody for IP, and I'll cover more some of the products we have for that area. And then we will finally finish off with some best practices to consider for IP. This includes basically setting up good controls, as well as how you can maybe troubleshoot common problems that might arise during your experiment. So immunoprecipitation, or what we refer to as IP for the rest of the talk, right, is essentially a technique where you are attempting to isolate a protein of interest from a cell extract using an antibody that's going to bind to that protein of interest. The IP itself consists of 5 basic steps, okay? First, we prepare our lysate, okay? So you have to lyse your cells, get the proteins out of solution. Then you will incubate your lysate with your antibody against that protein of interest. During this binding, the antibody at this point has usually been linked up with some sort of bead matrix. Very commonly, probably heard that protein AG beads are used, okay? So you incubate your lysate with this antibody bead complex. And after the incubation, you then do a series of washes to remove any non-specific binding material away from this basically tube, okay? After your washes, you then will do an elution that will help you basically remove the bound protein of interest from the antibody. And then you can analyze that eluted material on a gel as well as a Western blot, okay? The gel will allow you to visualize if any protein is actually present and basically verify that the IP worked. And then the Western blot, right, you then can look at the protein expression, right? You can look for the protein of interest, or you can also probe for associated binding partners if maybe you're doing a co-IP. So now we'll get into each of the steps of the IP in more detail, all right? So I mentioned the first step, right, is preparation of that cell lysate, right? You can't just apply an antibody to your cells in cell culture, right? It won't reach your target. You have to lyse your cells. The proteins can basically come out into the solution, be released. So the lysis buffer you're going to use for your cells, of course, is very much going to depend on the cell type itself, okay? For example, plant cells will probably require a very different lysis buffer than mammalian cells. If you're more interested in finding out some of the exact lysis buffers to use for these cells, we do have detailed protocols for different cell types on our website that you can refer to later. But in general, for a successful IP, we usually recommend about 500 microgram of protein, which could probably be isolated from either around 1 million mammalian cells or about 10 grams of plant material. When you are preparing your lysate, it's very important for the IP to always use fresh lysates. We don't want to use frozen lysates because when you do freeze thawing, that can result in potential protein degradation, aggregation, or perhaps even buffer instability. And as a result, you may not get as great of an IP. So after we have prepared the lysate, we then proceed to the binding step, okay? That is when we incubate the lysate with your antibody, nanobody, or whatever detection reagent, right? That's been attached to some sort of bead matrix. When you're doing the incubation, there are a couple parameters to keep in mind, okay? The time is very important. Most high affinity, good antibodies, you'll need to really incubate for about an hour. If you're maybe working with like a lower abundance target, you could increase the incubation maybe to 4 to 6 hours, give the antibody more time to bind the target. However, we don't recommend doing overnight incubation because the longer you go, the more you might increase the potential, right, for maybe non-specific material binding to your antibody. Also, with overnight incubation, you also run the risk of maybe your proteins or even your antibodies beginning to degrade. So try to stick to 1 hour for most targets. While you're doing the incubation, it's also very important that you're keeping those, right, those beads that the antibodies are attached to in a suspension, right? Because they're beads, they're going to have the tendency to want to settle to the bottom of the tube, okay? So to prevent that settling, we always try to mix kind of an end-to-end suspension, okay? That will keep everything kind of flowing and will basically promote this interaction with the antibodies and the protein of interest. The volume that you use for basically your lysate material also matters. Generally, 200 to 500 microliter is sort of that sweet spot. If you have too large of a sample volume, that runs the risk of your sample being maybe too dilute, and then your antibody will have trouble detecting your protein. Conversely, if you use too small of a sample volume, you then run the risk of evaporation and potential sample loss. Always carry out your binding sort of at 4 degrees. Remember, this antibody protein binding is simply a chemical reaction. And so for those kinetics to properly occur, we want to have the right temperature conditions. At 4 degrees, that sort of ensures optimal binding of the antibody and the protein. Next, after our binding, we then move on to the washing. And the washing is very important because remember, it's lysate. It's not just your protein of interest. It's all other proteins that are sitting in the cell. So we want to get rid of that non-specific material. Otherwise, they're going to have a very dirty gel downstream. So for the washing, we do basically, we apply washing buffer, and we either are going to spin down the column or use magnets to basically settle the beads or have the beads stick to the sides of the tube, and then we can discard that supernatant, or which would be our flow through. So when you are washing, some things to keep in mind. Your wash buffer should be harsher or stronger than your binding buffer, okay? Because remember, the purpose of the washing is to dislodge any non-specific material that may have also bound to your antibody or your bead beyond your protein of interest. So if you use, basically, you increase the salt or detergent concentrations in your wash buffer, that will help to disrupt those kind of bonds that are being made with the non-specific material. The timing for the washing also matters, right? Remember, these are still harsher buffers, right? So if you wash for a prolonged amount of time, that then will also start to disrupt the binding between the antibody and the protein of interest. So it's important to keep your washes short, and do not excessively repeat the washing either, again, because the more you wash, then it will become too much also for the antibody to withstand. After our washing, right, after we've removed any non-specific components, we then can proceed to elute our protein of interest from our antibody. And so you can do elution one of three ways. A lot of people may do denaturing elution using an SDS buffer. This is probably the strongest buffer, right? Because it's going to completely denature your protein, both the antibody and the protein of interest, which allow your protein of interest to be cleaved off from the antibody, then you can analyze in the gel, okay? But there are certain applications where we want to preserve the protein more of its native state. So then we might do an acidic or a proteolytic method. So with acidic elution, you're going to use like a glycine buffer, and the acidity of this buffer essentially will disrupt the interaction between the antibody and the protein, and will allow for the protein basically to denature, allow for the protein to basically release in more of a native form. Finally, if you are working with tag proteins, you also can attempt to cleave off the tag from the protein of interest by using like a TEV protease, okay? So using a protease that's specific for the tag itself can help you release the tag from your protein of interest. And so basically your tag will remain bound to the antibody, and your protein then will be basically cleaved off, and that can be eluted, again, in a more native form. But of course, to use proteolytic elution, you have to be working with a tag protein. So finally, after we have eluted the protein from the antibody, we then can look at the results, okay? So typically, we analyze the eluted material in two ways. We first run a gel. Again, a Komasi stain here is just showing us if any protein is present, right? It's a quick and dirty way to just verify that the antibody, that the IP worked, right? If you see a band in your basically bound or elution fraction, that is proof that we pulled down something, right? But to make sure we pull down the right thing, we then have to do a Western, right? We will use an antibody against our protein of interest to verify that that is indeed the right protein. Additionally, if you're doing co-IP, where maybe trying to pull down more than one protein, you might then actually do a Western for the potential binding partner of your protein of interest to basically verify and prove maybe a hypothesis that these two proteins are interacting together. So that's all the steps of the IP. Now let's talk more about, exactly what antibody should you use for this process. Okay, so when you're picking an antibody for IP, there are a couple things to look at. One is, I would always recommend that you use an antibody that's already been validated for IP, preferably by the manufacturer, right? Because if the manufacturer is validated for IP, that means they have done that testing in-house. They're going to provide you with suggested dilutions to use for many different cell types, and maybe even a protocol. So it can kind of help to get your experiments going, can help you also with that optimization work. If you were to use a non-IP validated antibody, again, that's going to be more time in your hands trying to figure out how can I get the antibody to work for IP. And there's a chance that it might not work at all, right? So it's not only a waste of time, it's going to be a waste of money. Make sure the antibody you choose has a high affinity for your protein of interest. This is especially important if you're working with low abundance targets, because again, if you have a poorly expressed protein in your sample, you're going to want the highest affinity possible detection tool to enrich for it in your sample. The sort of antigen you're going to have your antibody against, again, it's going to really determine, it's going to be based off the protein you are working with, right? If you are working with tagged protein constructs, you may want to then use, instead of a antibody against the PLI, an antibody against that tag, right? You can use a GFP or flag antibody for the GFP or flag tag and basically pull down the protein that way. And then, of course, your choice of binding matrix is going to really depend upon the type of protocol you wish to follow for the IP. So I mentioned during the washing, right? You can sort of wash either by spinning down your tubes, let the beads settle, or you can use magnets to have the beads adhere to the sides. So if you prefer doing IP with centrifuges and spinning down in spin columns, we would then recommend like maybe an agarose bead type substance. If you prefer to use magnets for IP, then you may want to use like magnetic beads or magnetic particles for those wash steps. So here at Proteintech, I do want to point out that we have over 4,800 antibodies that we have already validated for IP in-house in a broad range of cell lines and tissue types, okay? I mentioned that it's really important to use an IP-validated antibody for your IP experiment. So if you come to us, we have a lot of great products that we can help you with for this protocol. We have a broad range of IP antibodies against cancer, immunology, neuroscience targets. The chances are we might have what you're looking for. And our IP antibodies, they come in a wide range of formats. The format you choose probably is going to be dependent upon the target you're studying, as well as maybe how much you want to get out of your antibody. So if you are working with, let's say, a lower abundance target or a poorly expressed protein, we would recommend using one of our polyclonal antibodies, right? Because by their nature, polyclonals recognize multiple epitopes of a target protein. So it's going to increase your chance of being able to pull down that protein of interest. If you have more of a highly expressed protein, you know it's expressing well in your sample or tissue of interest, then you can go for one of our monoclonals or recombinants. So these are high affinity antibodies that recognize a single epitope on the protein. And if you're looking for something with more, an antibody that can do kind of more than one thing, maybe you want an antibody that's going to work well for IP, Western blot, immunofluorescence. We have recently launched a new line of primary antibodies called multi-Rabs. So these are basically a mix of clones that recognize multiple epitopes on your protein of interest, but these clones have all been recombinantly expressed. So you're getting the benefits of both a polyclonal and a recombinant all in one. Outside of our antibodies, we also have a pretty wide catalog of nanobody reagents that can be used for IP. And we will get more into the next section of where these nanobodies might actually come in handy for IP compared to an antibody. But in general, these are the two types of nanobody products we have for IP. So we have our nanotraps, okay? These essentially are affinity resins that consist of a nanobody that has been pre-conjugated to either an agarose or magnetic agarose bead. And these nanobodies that we have in the traps are primarily against fluorescent protein or peptide epitope tags. So if you are working with any tag protein constructs that include tags like GFP, RFP, HIS, MYC, HA, V5, we have great affinity resins that can be used to pull down those tag proteins from your sample. Beyond the tag protein products, we also recently launched a new class of products called anti-IgG VHH beads. So these products are sort of an alternative to protein AG. As I will show in the coming slides, there are a number of issues that arise when you use protein AG beads. Namely, you get a lot of protein AG contamination these IgG VHH beads can be used in place of the protein AG bead to basically reduce that contamination in your data. So you get much cleaner results. So we have made these IgG VHH beads against rabbit and mouse IgGs, as well as the subtype specific resin. So if you have like an IgG1, IgG2A primary, you can use our beads to basically pull down that type of antibody specifically. So now let's kind of get into more the advantages of using a nanobody versus an antibody in IP. So first, we will start with our nanotraps, okay? As a reminder, the nanotrap consists of a nanobody or VHH that has been pre-conjugated to an agarose or magnetic agarose bead, okay? It is a ready-to-use resin, meaning that you don't have to incubate the nanobody with the bead separately to get that conjugation to occur. It's all even linked to the bead. You just simply have to incubate the trap with your sample. So here we have our GFP trap, which basically is our nanotrap for pulling down GFP tagged proteins. And on the left, you are seeing a pull down completed with our GFP trap. On the right, we have a pull down done in the more conventional manner, where we used a GFP antibody linked to a protein AG bead. I think what you can notice right away is that when I do a pull down with the antibody, I am seeing 3 different bands, okay? We've got the protein of interest and the heavy and light chain, okay? So there's a fair amount of contamination. And the reason this happens is because this interaction with the protein AG and the IgG, it's just affinity driven. It's not like cross-linked or covalently bond. So when I elute, that buffer is going to be strong enough to basically also cause this antibody to leach off of the bead along with your protein of interest, giving us 3 bands. Now, this might not be a problem for a lot of you, but if your protein of interest is the same size as the heavy or light chain, it is going to lead to difficulty interpreting the results. Meanwhile, if you use our GFP trap here, notice you're getting just 1 clean single band, okay? And that's due to the fact that our VHHs are covalently bound to the beads. So no matter how harsh of an elution buffer you use, the VHH will not come off. You will simply just be able to elute your protein of interest, and you'll get much cleaner results. Furthermore, we have found that a lot of our nanobodies have much higher affinity for the protein of interest compared to antibodies. When we look at our Western downstream of our gel, look at protein expression, we see that with our nanotrap, all of your protein of interest is in the bound fraction. Nothing was lost in the flow through. So it was successful in pulling everything down. When I look at the Western blot using the antibody pull down, notice how the antibody was not as effective pulling down all the GFP protein. Some of it was lost in the flow through. And this is probably going to be a problem, again, if you're working with the low abundance, poorly expressed protein, right? You're probably going to want to enrich as much as possible. And if you're losing some of it, that's just going to give you less protein then for downstream work. So that's sort of showing our nanotraps. I mentioned we have another nanobody product, right? Our anti-IgG VHH beads, okay? And these are, as a reminder, can be used in place of protein AG to pull down really any protein of interest, okay? So how this works is like the trap, it's also a nanobody conjugated to an agarose or magnetic agarose bead. But in this case, the nanobodies are raised against either rabbit or mouse IgGs, okay? So similarly, how do you use a protein AG bead? You would simply incubate your antibody with these beads, okay, to form that matrix, and then you can use that for the pull down. So here we have a pull down of pCNA protein, okay, using either our anti-IgG VHH beads or protein AG beads from various competitors. What you will see is when I do a pull down with the protein AG bead antibody complex, we have the issue of basically the protein AG leaching off this bead and then showing up in the gel. This is a common problem. Like the protein AG is simply just coated on this bead. It can come off pretty easily. And when that happens, the protein AG then in your gel, in basically your elution fraction, can easily then also end up binding to your detection antibody using for your Western, or can bind to the secondary antibody in the Western. And so as a result, you're seeing multiple bands in my Western blot when I use a protein AGB. And notice how some of these bands are pretty close in size to the pCNA protein. So let's just say if this protein was not as highly expressed, you might have difficulty in figuring out, did I actually pull down the right thing? Meanwhile, if you use our anti-IgG VHH beads, we're just seeing one clean band of the pCNA, okay? Because again, our nanobodies are covalently bound to this bead, they're not going to come off. So you're not going to get any additional contamination like the protein AG. And then here, we're just illustrating what you would see in your protein gel downstream of the IP, right? So if you do an IP with either our IgG bead, okay, after the elution in your chromatography gel, you'll probably see 3 bands, right? You're going to see your protein of interest and your heavy and light chain, right? Because whatever is bound to your nanobody will come off. When I use a protein AG bead for the pull down, not only am I going to see my protein of interest, heavy and light chain in the gel, but I'm also going to see several additional bands, which is likely that protein AG that came off the bead, okay? And as I just showed in the previous slide, when I do a Western on this fraction, you're going to you run the risk of your detection antibody binding these other non-specific components, giving you other bands or smears. So here's another example, right? In this case, we're pulling down a Bax protein using either a rabbit anti-Bax antibody or a mouse anti-Bax antibody. And then we have done the pull down basically with that in conjunction with the anti-rabbit IgG beads or the anti-mouse IgG beads. Again, take a look. When we use our anti-IgG beads, we get one clean band for the Bax protein. If I'm using the protein AG beads, we're seeing a high background, right? Because as I mentioned, protein AG can indeed dissociate or leak from the beads upon the elution, causing contamination in the protein of interest. And remember, the protein AG can also bind non-specifically really to any detection antibody you might be using for your blot, which is why this shows up on the Western blot, in addition to your protein of interest. Additionally, our anti-IgG beads, I mentioned, right, they're much cleaner results. They don't leach off the bead, but they're also really specific for their IgG of interest, meaning that our rabbit IgG bead only binds rabbit IgGs. Our mouse IgG beads only bind mouse IgGs. They don't bind non-specifically to any human or bovine IgGs, which might be present, right, if your samples contain HSA, FBS, BSA, etc., right? We know that BSA and HSA are commonly used as stabilizers for a lot of antibodies, and most cell culture media usually has FBS in it, right? So when we are then running our IP from these samples using these antibodies in question, right, there's a small risk that if using the protein AG may end up binding to any human or bovine IgGs present in your sample. And indeed, what we see here is when we did a pull down using our beads from either human serum or bovine serum, when you use our IgG beads, you get a clean band, right? They do not bind non-specifically to any human or bovine IgGs. But if I use protein AG, we are seeing a huge mess, like several bands here, like they're binding to all sorts of human IgGs in particular, right? So you're getting much higher specificity if you use the anti-IgG VHH beads. So to kind of summarize what would then be the best product for you to use for your IP, right? Based on the kind of three different products I've covered, either antibodies or antibody reagents. If you are working with any sort of tag protein, you're working with GFP tags, RP tags, V5, MYC, HA, strep. Remember, we have a great range of nanobody-based reagents called the nanotraps that work exceptionally well for the pull down of tag proteins, right? So you can use our nanotraps on the tag proteins to get clean single band pull downs without any heavy and light chain contamination. Because they're also pre-conjugated, ready to use, that's going to save you also significant time on your IP. You can get an IP done with nanotraps in like less than 2 hours. Now, if you are perhaps working with endogenous proteins, okay, and would like to have a cleaner alternative than protein AG for pull down, then we are going to recommend, right, our anti-IgG VHH beads, right? You can use these in place of the protein AG bead to pull down any rabbit or mouse primary IgG, okay? As we have covered, they're highly specific for their target IgG. They're not going to bind non-specifically any mouse, any human or bovine IgG, and you get much cleaner results, right? These nanobodies do not come off of the bead like the protein AG commonly will during the elution step. So now we've outlined the reagents. I think it's time to cover some best practices, right? What are some controls you should kind of establish when doing an IP? How can we troubleshoot experiments that are going wrong? So starting with the control, I think all of any basic experiment, you want to have a positive control, right? So in the case of IP, your positive control would be helping you basically verify that protein enrichment is actually happening. Okay, you want a control that's for sure is going to work. So you can use a couple different things as controls. If you are working with a tag protein, you can simply just run the tag only, just in your sample. That would be a great positive control. If you're maybe working with an endogenous protein and you happen to have access to a purified recombinant version, you can just run that as a sample, okay? And then you hopefully be able to get a nice band. If you are not doing a tag protein or you don't have access to a recombinant protein, there are other options used for positive controls. You could either get a sample from a tissue or cell line where this protein interest is highly expressed and run that. Or if you have the construct handy, you can also overexpress it in your cell to basically, again, create that positive signal for your control. Conversely, you also want to have 2 types of negative controls. Your first negative control is you want to basically have a control to verify that your antibody is not binding to any unspecific material, okay? So for your negative control to verify the antibody specific, you simply can do one or two things, okay? If you're working with a tag protein, you can try running a sample of the protein without the tag, okay? So if you're using an anti-tag antibody or an antibody for the pull down, if you have a tagless protein sample, it should, in theory, bind nothing, and that would be a good negative control to use. If you're working, let's say, an endogenous protein, you also can do a knockdown. You can knock out the expression of the protein in your cell and then run that as a negative control. Your second negative control you want to have is you want to be able to also verify that your beads, right, whether they're agarose, magnetic beads, or particles, you want to make sure that bead matrix is also not non-specifically binding anything, right? If your beads are binding a lot of garbage, it's going to give you kind of messy results. So for a negative bead control, we can do 1 of 3 options. We can just try running a sample with essentially blocked beads, basically beads that don't have a nanobody or antibody attached, right? If those beads, you see the sample with the bead, it binds a lot of material, and the beads are not very good. You can also attempt to pre-clear your sample. So what you could do is you can incubate your sample beforehand just with the beads before the IP, and then run that fraction. And that fraction in theory should remain pretty clean, right? Or if not, right, the pre-clearing could basically help to remove any of the non-specific material that might be present, and it can correct for the background. And finally, you can also try doing isotype controls. So now let's look at how can we best troubleshoot an IP experiment. So I'm going to put out one example here of an IP, and we're going to look at a couple things that might go wrong and how we can correct it. So for our example here, that you're going to see in the next couple slides, we have essentially taken a GFP protein, a GFP tagged protein, that we have pulled down with our GFP trap product, and then we are doing a Western downstream, the GFP antibody, to look at the protein expression. So one issue that might arise is, let us say that we have done the IP, we did the pull down with the GFP trap, but the problem is we end up seeing a lot of background in the bound fraction. If you look at your gel and your Western, there are many bands in addition to the protein of interest. This obviously is not ideal, right? I mentioned the traps, right? One of the biggest selling points we have is that they're supposed to give you clean single band pull downs. So if you're seeing this, what is the reason for that? And it could be one of two things. It might be that your cell lysate is too highly concentrated, right? If there's just too much cells or protein sitting around, that does increase the risk, right, of there being, of course, naturally more non-specific components that might stick to your resin. The other issue could be that maybe your protein of interest, it got degraded, right, in the sample somehow, which is why, especially on the Western, you're seeing multiple size bands, right? Maybe your protein of interest is clumped up, so it's showing up as larger than normal, or it's degraded, showing that it's smaller than normal. So to kind of correct for these issues, we can try looking at a couple things, right? If your sample is too highly concentrated, you may, of course, want to consider, when making the lysate, perhaps diluting your sample more, right, to help wash out some of those non-specific components and give you a cleaner sample. Remember, during the binding step, right, the incubation, there are a couple parameters to keep in mind to reduce for that non-specific binding, right? Incubation at 4 degrees is critical, right? If we incubate at a higher temperature, that does run the risk of the antibody protein binding interaction not happening optimally, and you run the risk of either A, your proteins degrading, or maybe your antibody picking up more garbage. Again, the same risks also arise if you over incubate, right? The longer you incubate, you're then just giving your antibody more time to end up maybe picking up stuff that is not your protein of interest. We also can attempt, right, to pre-clear the sample, right? So before doing your pull down, you can incubate your sample with blank beads to perhaps remove any potential background. And then if you are seeing degraded protein, right, it could also be that perhaps your protein was degraded by an endogenous protease in the sample, okay? Remember, when you lyse a cell, you're releasing all the proteins out, including those proteases. So if you want the proteases to not chop up your protein before you can get to it, add protease inhibitors to your lysis buffer immediately after to kind of prevent that. And then washing, there are a couple of things we can consider here too, right? Remember, you want to use a kind of pretty strong washing buffer to, again, dislodge and remove any non-specific components that may have attached to your resin during the binding step. If I use too gentle of a wash buffer, you do run the risk of, right, additional materials not getting properly washed away. So that is the issue of high background. Now, what if I do a pull down with my GFP trap, but I see nothing in the bound fraction? I didn't pull down anything, right? If you notice in your input and flow throughs, we do have protein in the gel. When I do a Western, I am detecting the GFP in the input and flow through, but I'm seeing nothing in the bound fraction. So what happened there, right? It's probably an issue that your, in this case, this nanobody didn't probably bind enough or any protein at all. And that could be due to a couple things. It might be that our protein of interest, it degraded, it aggregated, it became insoluble for whatever reason, so we couldn't bind it. Perhaps the mixing was not thorough enough, right? If maybe you were not going end to end, just to let the nanobodies settle, they're not going to bind to anything. It might be that we did not apply enough slurry, we didn't add enough basically nanobody bead slurry to our lysate, so there wasn't enough basically to pull down the protein. Or maybe it's that our protein of interest actually it's so large and clunky, it couldn't properly attach to our resin. So if you want to correct for these items, again, the lysate prep is very important, okay? You want to use a good enough lysate buffer that will ensure your proteins can stay in solution. If it's not strong enough, maybe your proteins get lost in the pellet when you wash the sample down. Again, during the mixing, we want to do end over end rotation, right? Because that prevents the beads from settling to the bottom of the tube and also promotes nice, even interaction between all your antibody and your protein of interest. In the case of our nano traps, we recommend using 25 microliter of slurry for every IP reaction. If you're using antibodies, right, you're going to have to probably follow the protocol you suggested dilutions, but kind of use at least the minimum amount required, recommended by the manufacturer, because that just shows that that's what they have tested the product to work at. And then in the case of maybe your protein complex being too big that it can't properly attach to your resin, you could try switching up the bead matrix, okay? So for example, with our nano traps and our IgG beads, right, we have these conjugated to agarose and magnetic agarose. These are both porous substances that are coated both internally and externally with nanobody, meaning they can bind in theory a lot more protein. But if your protein is too big, it won't be able to get into those pores. And so to correct that issue, we also offer a lot of our traps conjugated to solid magnetic particles that are only coated externally with the nanobody. And so that will allow for larger complexes to better align, okay? So let's say if you're working with antibodies, then maybe you want to try, say, similarly, maybe switching from a porous bead to, like, let's say, a dynabead, something that's solid, that you can only coat externally with your antibody to better capture those larger protein complexes. Next common IP issue, right? In this case, I did my pull down, but when I do my Western, I am unable to detect any protein of interest in any of my fractions, okay? So it's not only is it not here in the gel, but it's also completely invisible in the Western, okay? So if that is the problem, it is likely that the amount of protein in your sample maybe is just too low, right? That nothing will be pulled down and naturally nothing will be able to be detected either. Or it might be an issue that your protein is insoluble. When you do the lysis, it just crashed out and then it ended up disappearing in the pellet. So if we want to correct for that, okay, if you have, let's say, a poorly expressed protein that's not showing up, you may need to alter the protein expression method. Let's say if it's a fusion protein, maybe you have to optimize your transfection to get it to express better. Or, right, you could also try using more cells or concentrating your lysate more to correct and ensure for more protein being present in the sample to be pulled down. The other issue that maybe you're not detecting proteins, maybe you actually are pulling down protein, but perhaps just your Western blot antibody is bad. It's unable to detect the protein in the gel. So if that is the problem, then you may want to play around with the antibody or try other antibodies to get a signal. Our final example here is, let's say I have done my pull down and I have a band, I successfully pulled down my protein of interest, but when I do my Western, I'm noticing that the expression level in my bound fraction or my eluted material is far less than the expression in my input, okay? That is kind of the opposite, right? We generally, for a good IP, should be seeing more protein in your bound fraction because it's been enriched for, not less. And so if that is the problem, that could be that the nanobody in this case may successfully bound the bead, but your elution was bad. Your elution buffer may have not been strong enough to actually release the protein, and so a lot of it still remains stuck to the actual resin. So if we want to crack for that, right, you want to then use a stronger elution buffer. SDS, right, is as strong as you can get. It's going to completely denature your protein, allow it to release. If you are not concerned about preserving the protein in its native state, this will help you get everything else. So that basically brings us to the end of this webinar. Just some things, again, to keep in mind after everything I've said, right, is the choice of antibody or nanobody for your IP is, of course, very important based on what you're working with and what kind of background you're concerned about, right? We have different tools that can help you to correct for all of those issues. We have already reviewed how sample prep indeed affects your results, how also in every step of the IP, there are also specific parameters to ensure for in order to get the best results. And of course, whereas for any experiment, you want to have good controls, okay? And these controls, in turn, can also help you troubleshoot your results if things go wrong. So with that, I will conclude and hand it back off to Poppy for any questions.
Thank you so much, Rahul. That was really clear, really great walkthrough through the whole workflow. We have had quite a few questions come through as well, but if there are any more, please everyone do remember just to put them in either the chat or the Q&A box. Just before we come on to those, I will just share a closing slide with you all. So if you found this useful and you do want to learn more either about immunoprecipitation or about any other immunoassay application, we have lots of other videos like this, some more specific in our video hub you can find on our website. We also have an online learning portal that you can access at learn.ptglab.com, which has several completely free online training courses that you can walk through at your own time. These cover not only immunoassay applications, but also things like statistics and coding. And finally, we also have tomorrow's webinar, don't forget, on ELISA at the same time. If you're not registered, there's still time to register. And yeah, just to answer all the questions in the chat, we will be sending out all of the recordings from all of this week to anyone who's registered. So don't worry about receiving those, but please do allow up to a week to receive them. And just something else to bring to your attention, everybody who has registered and attended one of these webinars this week is able to get a 25% off one full-size primary antibody with the promo code antibody25. But again, all of the T's and C's of that will be sent out in the follow-up email, so do just keep an eye out on your email. If you've got any other questions, you can direct those towards events@ptglab.com, or if you've got any specific immunoprecipitation questions or product questions, you can reach Rahul directly at his email just there. And with that, I will just stop sharing and come on to some of these questions. So a couple of people actually have asked whether or not it's better to mix the lysate and the antibody first and then add that to the beads, or is it better to add the antibody and the beads at the same time as the lysate?
So I think it doesn't matter, right? For example, like our traps are ready to use, pre-conjugated. So you're going to be adding essentially the whole thing to lysate right away. So even if you're working with an antibody, you can kind of do either method. Just remember, I mentioned that when it comes to the incubation, you don't want to incubate for too long. So for some reason, it takes your antibodies and your beads a long time to attach. Maybe you want to do that separately before the incubation step. But otherwise, the order doesn't quite matter.
Okay, perfect. Thank you. And someone has asked whether or not, if they've created a cell lysate and they've ended up with quite a gloopy cell lysate, will that affect the binding of the antibody? And do you have any advice to help reduce like the sort of the viscosity of their sample?
I would think you would need to dilute that a little more. It's probably just too sticky. But we also know, for example, plant extracts are notoriously sticky, kind of like that. And then you might just have to, when you wash, use a harsher wash buffer to get rid of any of that stickier material.
That's great. Thank you. Just to add to that as well, I know that I definitely had issues with sometimes getting really viscous lysate if there was a lot of DNA present. So you can sometimes try and help reduce that by either adding in something like DNAs or just by sonicating your sample. That can sometimes help as well. So a few other questions then. Someone has said, can you reuse your antibodies for downstream Western blot application?
I mean, you can use the same antibody both for IP and Western. That's not an issue. I think generally what we see a lot of people do is they might use different antibodies, specifically if they're doing stuff like co-IP. You might use, right, one antibody for your protein of interest, and then a different one to look at the potential binding partner.
Great, thank you. So a question here from Sophie. If you want to analyze your IP by LCMS, is it better to freeze replicates before the IP or after IP to analyze them all at MS at the same time?
That is a very good question, but might be out of my scope of expertise. But we can relay that to our R&D team and try getting back to you.
Yeah, definitely. And if as I say, if there are any other questions as well that we haven't made it around to today, just throw them in an email and we can get those directly to our scientists here at hopefully be able to answer those. So sorry, there are so many questions, but I'm just trying to find one that we haven't answered yet. Somebody has asked, is the efficiency of the pull down affected by the size of the protein?
It can be, right? So remember I mentioned that if you're working with a very large protein complex, so we're talking probably like in the mega Dalton range. Such a clunky complex, right, might have trouble sticking to an antibody or an antibody if that's in a porous bead and it's within the bead itself. It won't be able to get into the pores. So in that case, if you're maybe using a porous bead to pull down a very large protein complex, you might see perhaps a inefficient pull down, maybe a lot in your flow through because not everything could probably properly adhere to the bead, right? So to correct for that, you may want to do solid bead, right, which is only coated externally. So there is more space for those larger proteins to attach. But for most researchers, this is probably not going to be a problem. You're likely not pulling down something that huge or massive. But if you are, right, there are ways to work around that.
Okay, awesome. But it sounds like that tends to be more of an issue only when you're looking at large complexes as opposed to
Yeah.
just a slightly larger 200 kilodalton, say, protein. Okay, so somebody has asked about elution of the GFP traps. And they've said for magnetic GFP trap in either SDS or acidic elution, is it necessary to heat at 90 to 100 degrees, or is the buffer alone enough of eluting it?
I think the buffer should be fine. I'd have to double check our protocol and get back to you, but these protocols are also on our website. You can look at our GFP trap page, and the protocol has pretty detailed elution instructions.
Yeah, I think whenever it's a specific product related question, it's always best just to check the product data sheet, or the information will be on there. But I agree with you. I do think it's fine just to use the buffer alone. Okay, maybe looking at the time, we've got time for one, maybe two more. One says, do you have any specific recommendations for co-IP using GFP trap analyzed by mass spec? Is that something that we know has been done before?
I believe it definitely has. We do have an on-bead like digest protocol that can help prepare your samples for mass spec. Again, I'd refer to the protocol for more specifics.
And some more questions about the magnetic GFP traps. Are they reusable?
Generally, no. Um, because again, this has to do with the nanobody has really high affinity for the protein of interest. So you generally have to use a stronger elution method to elute your protein off. And after that elution, the nanobody will probably lose a bit of its functionality. So if you were to try to reuse that resin, you probably will not get as good of a pull down. So usually do not recommend reusing the traps more than once for a pull down.
Great, thank you. And then we've had one final question come in, so I think we'll end on this one. This person is saying that their protein of interest does not show a detectable band in Western blot in normal conditions, but it shows a clear band when they overexpress. So they'd like to use IP to study the endogenous protein. What are the best strategies or optimizations to successfully immunoprecipitate? I guess quite a low level expressing protein is what they're asking for.
Yeah. So again, I mentioned our traps work really well for low abundance proteins. So if you are able to attach a tag to the protein and then attempt to express it, that would probably do a good job in pulling things down. Again, you could also probably try our IgG beads, right? If you just need a higher affinity antibody, you could try also other antibodies out there. But yeah, see if you can attach a tag to your protein and go that way.
Great, thank you. We've had, as I say, lots and lots of comments saying how useful it's been. So really appreciate your time. Thank you to everybody who has attended this webinar or another one this week. Please do keep an eye out on your emails. You'll receive all of the recordings probably early next week, along with all of your individual certificates of attendance. And with that, I'm going to bring the webinar to an end, but I hope you all have a great rest of your day.